Hello, and welcome to today's webinar, Air Pollution and Breast Cancer, Evaluating Geographic Variability and Underlying Pathways. I am Dr. Barbara Malkus, Board President for Massachusetts Breast Cancer Coalition, and I'd like to thank you all for joining us today. Massachusetts Breast Cancer Coalition is dedicated to preventing environmental causes of breast cancer through community education, research advocacy, and changes to public policy. I am pleased to welcome today's speaker, Dr. Alexandra White. Dr. Alexandra White is a Stadtman Investigator in the Epidemiology Branch in the Division of Intramural Research at the National Institute of Environmental Health Science, where she leads the Environment and Cancer Epidemiology Group. She received her MSPH in 2012 and her PhD in 2015 from the Department of Epidemiology at the University of North Carolina at Chapel Hill Gillings School of Global Public Health. Dr. White is a co-investigator of the SISTER study, a large prospective cohort of women in the US who are followed for breast cancer and other health outcomes. Her research focuses on identifying environment and lifestyle risk factors for breast and reproductive cancers and understanding the biological mechanisms that allow cancer to develop. Dr. White uses geospatial data to study widespread environmental exposures, such as air pollution, and focuses on cancer risks associated with chemicals found in personal care and other everyday products. Dr. White, welcome.
Thank you so much, and thank you for that kind introduction and for the invitation to be here today. I'm really excited to have the opportunity to share the work that we've been doing with the SISTER study and in my research group, and I look forward to also some discussion at the end. So today we're going to be focusing on talking about outdoor air pollution. Outdoor air pollution is a very complex mixture that we all experience, and this exposure is ubiquitous. It is complex in the fact that it arises from a variety of different sources. We're exposed to air pollution from industrial sources, from traffic, as well as for emerging threats such as wildfires. The International Agency for Research on Cancer has classified outdoor air pollution as a Group 1 carcinogen, and this is their highest level of classification in terms of their confidence in stating that this acts as a carcinogen. But this decision was primarily based on evidence for fine particulate matter, or PM2.5, and lung cancer. And there's much less evidence for how air pollution may be related to other cancer types. But I think it's biologically plausible that air pollution may be related to the risk of hormone-sensitive cancers. Air pollution contains chemicals that may act as carcinogens, but also as endocrine disruptors. And so endocrine disruptors might be particularly relevant for something like breast cancer, which has a hormonal component. We don't typically think of air pollution as being relevant for the breast, but studies show that compounds we inhale enter our circulation and reach all organs, including the breast tissue. Most previous studies of air pollution focus on nitrogen dioxide, or NO2, which is considered a proxy for traffic, as well as fine particulate matter, PM2.5. And breast cancer is important because I know this audience is familiar with this, but it is the most common cancer diagnosed among women in the US and worldwide. It's estimated that there'll be over 300,000 new cases diagnosed in 2025, and this translates to 1 in 8 women being diagnosed with breast cancer at some point in their lifetime. Breast cancer is a heterogeneous disease with both risk factors and survival after diagnosis varying based on tumor subtypes. We often define tumor subtypes using the hormone receptors estrogen and progesterone. Hormone receptor positive tumors are more commonly diagnosed, whereas hormone receptor negative tumors are less frequent, but women who are diagnosed with those tumors tend to have worse survival after diagnosis. There are a wide range of established risk factors for breast cancer. Many of them have to do with a woman's reproductive history, including age at menarche, how many children the person has had, and time spent breastfeeding, as well as use of exogenous hormones. And these risk factors together really emphasize the importance of the hormones and the potential for the impact of environmental chemicals that may act as endocrine disruptors. So as I mentioned, previous studies have largely focused on NO2, which is a proxy for traffic-related emissions. A meta-analysis and systematic review in 2021 found that NO2 was associated with a slightly higher risk of breast cancer. In contrast, there's been less consistent evidence for fine particulate matter, with the same meta-analysis reporting a null association, although there have been some recent studies that have been consistent with a more positive association. But in general, there's very little evidence in how the relationship between air pollution and breast cancer varies by important subgroups. For example, we know very little about how associations might vary by breast tumor characteristics, including hormone receptor status. We also know that geographic variability could play a very important role. Fine particulate matter in particular varies across the US due to differing sources of exposure. So PM2.5 is called that because it's a measure of particles that are less than 2.5 microns in diameter. But that measurement of total PM2.5 really doesn't tell us about what types of PM2.5 people are exposed to. And this really does vary across the US. And so we've seen this in other studies that we've done. So in work I led in the Black Women's Health Study, which is a large cohort of Black women across the US, we looked at residential exposure and enrollment to PM2.5, NO2, and ozone. And although we saw little evidence of an association overall, when we looked at this association by the geographic region that the women lived in, we saw that among women living in the Midwestern US, there was a significant association between PM2.5 exposure and breast cancer overall. But there's a number of important limitations to keep in mind of the literature so far. In terms of air pollution and a range of health outcomes beyond just breast, but also cardiovascular disease, we tend to see relatively small associations. And so there's a real need to maximize our study power to make sure that we're able to detect these associations that, even though they may be small, they are still relevant on a public health scale, given the fact that exposure to air pollution is so widespread. Also, because it's challenging to look at this association within single studies, they often haven't had the power to consider etiologically distinct subgroups where associations may be stronger, such as by tumor subtype. Additionally, many studies have focused on assessing exposure to air pollution using a single time point, which is largely the time that a woman's enrolled in a study. And so this can be somewhat arbitrary, and it might not be the most relevant time period for exposure. For breast cancer, we know that there's a long latency for exposure. So exposures, 10 to 15 years prior can play a role in breast cancer development. And so just having exposure assessed at a single time point may be causing misclassification. So with that, for the first part of my talk today, I'm going to talk about a study that we've led evaluating air pollution and breast cancer using data from 5 large US cohorts. So this study builds on an established consortium that was designed to study air pollution and cardiovascular disease. And this work is led by Joel Kaufman's group at the University of Washington. And using fine-scale air pollution exposure models, they estimated continuous residential exposure to air pollutants over follow-up. So really, this is overcoming that limitation that I mentioned of just focusing on a single address, trying to better capture long-term exposure to air pollution. So for this analysis, we focused on the female-based cohorts listed here, the Nurses Health Study 1 and 2, the WHI Observational Study and Clinical Trial, and the Sister Study. Across these cohorts, we had over 400,000 women with an average follow-up of 17 years that we had estimates of their exposure to air pollutants at their home. And during this time, over 28,000 breast cancer cases were diagnosed. So we assessed exposure to NO2 and PM2.5 using these suite of regional spatiotemporal models. And what these models do is they incorporate a host of information from regulatory monitoring data as well as supplementary research monitors across the US. The model also takes into account a wide range of geographic covariates such as population density, traffic, green space, and this is all integrated to estimate a 24-month rolling average of exposure at the residence level. So jumping into our findings, so these figures show hazard ratios and 95% confidence intervals using that come from a Cox proportional hazards regression that has time-varying air pollutant exposure. So when the hazard ratios are greater than 1, that indicates a positive association and that this air pollutant is associated with a higher risk of developing breast cancer. So for NO2, we see evidence of a small, but consistent with previous studies, magnitude of association, where a 10 part per billion increase in NO2 is associated with about a 3% higher incidence of breast cancer. For PM2.5, we see little evidence of an association overall. But the real goal of the study was to get at these relevant subgroups because we pulled data across 5 large US studies to really give us the power to tease apart where these associations might be most relevant. So for NO2, we looked at how this association varied by a woman's menopausal status at diagnosis, as well as the hormone receptor status of the tumor. Associations for NO2 appeared slightly stronger for premenopausal women and those with hormone receptor positive tumors. We still didn't have a lot of power to explore differences by race and ethnicity, and we saw little difference by census regions. So we also looked at these subgroups for PM2.5, and here we do see an interesting association where PM2.5 is significantly associated with hormone receptor negative tumors. I think this is one of the most important findings of this study because these tumors are really difficult to study within a single cohort due to the fact that they are much less frequently diagnosed. These tumors, if you'll remember from how I discussed this before, these tumors also tend to have a worse prognosis. And so it's really important for us to identify new risk factors for these tumors. For PM2.5, we also saw some evidence of that geographic heterogeneity that we hypothesized. We see that among women living in the Midwest, that PM2.5 is also significantly associated with breast cancer in this population. And this finding is consistent with what we previously saw in the Black Women's Health Study. And so this suggests that PM2.5 in the Midwest might be particularly relevant to breast cancer. So to summarize these findings, this is a meta-analysis of newly developed data using information from 5 large US prospective cohorts. This is the largest study to date of air pollution and breast cancer in the US, and this gave us the ability to really evaluate those key subgroups. But despite using data from 5 large US cohorts, we still observe very wide confidence intervals for our race-specific estimates. For our exposure assessment here, we had time-varying exposure estimates over follow-up, which allowed us to consider a wider range of exposures. We also use the same exposure model across cohorts, which helps facilitate us pooling our data together. These are all very well characterized cohorts, and we have extensive information on other factors that may play a role that we were able to adjust for. We did a wide range of sensitivity analyses, including additional adjustments, such as those traditional breast cancer risk factors, and this did not change any of our conclusions. So moving forward, we were really interested in better understanding the importance of exposure temporality. Most studies really focus on relatively recent exposures, which may not be the most relevant. So one of the projects that we're working on now is evaluating the impact of historic exposure to air pollution and breast cancer in the sister study. So I want to start by giving a brief background on the sister study. So this is a prospective US-wide cohort. It includes over 50,000 women who were recruited between 2003 and 2009. All women in the sister study have a sister who was previously diagnosed with breast cancer, even though the participant in our study had no history of breast cancer herself. So that's the name, the sister study. So our participants, based on their family history, are at a higher risk of going on to develop breast cancer themselves. At the time they enrolled in the cohort, the women donated biologic specimen, had a home visit with a trained examiner, and completed extensive questionnaires. We continue to follow these women moving forward, so we collect information on their updated, any residential moves, as well as updated risk factor information. So to date, we have over 4,600 breast cancer cases that have been diagnosed in the cohort. So to give you an idea of the sister study participant characteristics, so at the time that they enrolled in the study, the women were on average about 55 years of age. The cohort is 9% Black, 5% Hispanic, and 84% non-Hispanic white. This is a slightly higher socioeconomic status cohort, so over half of our women have a bachelor's degree or higher, and a third have an annual household income greater than $100,000 a year. And this is consistent with other volunteer-based cohorts in the US. They tend to be higher SES. So in the sister study, at the time they enrolled, we collected information about their enrollment address, as well as we asked them about their longest lived adult address. As I mentioned, we also collect information on all of their follow-up addresses over time. And this is some of the data that I've already shown you, really focusing just on those follow-up addresses. But for all of our participants, we have these missing addresses, these gaps, based on whenever their longest lived adult address is. And so it's hard for us to reconstruct, going backwards, a residential history to estimate previous exposures. So we decided to move forward with a research project using data from a commercial database, LexisNexis, to build comprehensive residential histories so we could go back even further in time to see how past exposure to air pollution is related to breast cancer risk. So LexisNexis is a commercial vendor of data products, and it includes data from public records. So this can include mortgages, property deed transfers, credit reports, and a pilot study previously found that this is the address data from LexisNexis is the most complete and accurate compared to other vendors. In general, the data appears to be available starting around 1980, although there are some from earlier years. And so we aim to reconstruct sister study participants' residential histories using a complex data cleaning algorithm that incorporates information we already know, which I think was really important, that we had information on the baseline address and the longest adult address that was self-reported from our participants. And we could use LexisNexis to really fill in the gaps here. This data cleaning algorithm also removed short duration addresses and smoothed over time periods. So this effort was very successful. So for 96 of our sister study participants, we have complete residential history now going back to 1990. And for almost 3/4 of our participants, we can go back all the way to 1985. So this, our median, age at earliest address is about age 40. So for our participants, we're largely able to go back to about age 40. So once we built these comprehensive residential histories, our next step was to estimate annual average PM2.5 starting in 1980 and all the way through 2017, accounting for residential moves. And the goal here really was to capture how exposure to air pollution decades prior might be relevant to breast cancer risk and to identify potential susceptible windows of exposure. So I'm going to start by sharing some of our findings for NO2 as well. So in this analysis, we used an approach called distributed lag models, and these allow us to estimate an exposure lag response association and allows us to vary over time. So this is a very flexible modeling approach that allows us to investigate whether there are certain time periods that are more relevant. And so this creates a lag response curve, which is this graphical representation of the individual hazard ratios on how they contribute to overall breast cancer risk. So for example, in this figure, you can see that 0 is the year of diagnosis, and then 5 would be 5 years prior to diagnosis. So how does your exposure to NO2 at 5 years prior to your diagnosis, how is that related to your risk of breast cancer? This modeling approach also gives us an overall summary effect of the overall effective exposure history during this time. So here we see for NO2, we see that overall we have a positive association, about a 12% higher incidence of breast cancer for past NO2, which is stronger than what we've seen when we've focused on more recent exposure. And we see that in general, exposure in the last 10 years seems to contribute most. We also did this modeling approach for hormone receptor positive and negative cancer. The association for ER positive cancer is very similar, but for ER negative, we see little evidence of an association for past NO2. For PM2.5, we took a similar approach here using these flexible modeling, and we see something interesting, that's a little bit different. So for overall breast cancer, we see this. It seems about exposure 10 years prior is the most relevant for breast cancer risk. And this is really driven by those hormone receptor negative tumors. So we see the largest increase in risk is really for exposure, that's occurring more than 8 to 12 or 13 years in the past. And the summary overall hazard ratio for ER negative tumors is 1.57. So certainly it appears that considering the past exposure and the history of exposure really does strengthen the effects that we're able to observe. So in general, we saw evidence supporting that more recent exposure for NO2 may be related to breast cancer risk, and this is consistent with the hypothesis that certain types of air pollution may be acting through estrogenic or endocrine disrupting mechanisms and potentially acting as a tumor promoter. For PM2.5, we see this association with hormone receptor negative breast cancer, and our findings here suggest that exposure latency may be important. But a real limitation here is that we're unable to explore that PM2.5 heterogeneity that we've seen in our previous studies of that we've seen this geographic heterogeneity, suggesting that different types of PM might play an important role. So moving forward, we're also really interested in exploring potentially relevant underlying biologic pathways. And so to do this work, we've led a few studies evaluating the relationship between fine particulate matter and breast tissue characteristics using data from the Common Tissue Bank. So some of you may be familiar with this, but it's a very unique resource. It is a biorepository of normal breast tissue from volunteers. And at the time that we collaborated with them, they had over 4,000 breast tissue samples. And this is currently being led at Indiana. And so what we did is we took we estimated the annual average PM2.5 for the address that the woman lived at when she donated that normal breast tissue sample. And we did that using a linkage to the EPA CMAQ system. So our first study in this population, we looked at terminal duct globular units. These are sites where most breast tumors arise. So having more TDLUs indicates a higher risk of going on to develop breast cancer. And this was available in a subset of 2,000 women for which a trained pathologist had characterized these TDLUs. And what we saw is that women who lived in areas of higher exposure to PM2.5 had a higher TDLU count. So this suggests that PM2.5 might be impacting the environment of the breast tissue to influence a woman's risk of going on to develop breast cancer. And so moving forward, we were really excited by these findings, and we wanted to extend this to more comprehensive measures of breast tissue. And so this led to a collaboration with some investigators at the National Cancer Institute that were assessing histologic tissue characteristics in a larger sample of women in the Common Tissue Bank, almost 4,000 samples. And this analysis used a machine learning algorithm to look at these breast tissue slides and to quantify the percent epithelial, stromal, adipose, and total tissue area. In particular, we were interested in a metric called the percent epithelial to stroma, or ESP proportion, to kind of summarize all of these different metrics. And previous studies have shown that having a higher ESP, percent ESP, was associated with a higher risk of breast cancer. So to start, we looked at the same PM2.5 residential exposure at the time of donation and the association with the individual tissue metrics, as well as that integrated ESP measure. And so we saw that women who had higher residential exposure to PM2.5 had a higher percent adipose, a decreased percent stroma, a pretty null association with epithelium, and a slightly positive, but with confidence intervals that include the null association with ESP. But because we were really interested in understanding how different types of PM2.5 might impact this relationship, we also characterize women in the cohort based on their PM2.5 component profiles. And so what we did here is we identified subgroups of women that had similar PM2.5 components. And when we looked at the association based on these PM2.5 component profiles, we see that PM2.5 was associated with a significantly higher percent ESP among women living in a more Midwest urban cluster. And this cluster in particular was characterized by being very high in organic and elemental carbon, which are markers of traffic. And so it seems that, there is some evidence, the PM2.5 is impacting the breast tissue, and that it might vary by the types of PM2.5 that people are exposed to. But overall, we see that all types of PM2.5 seem to be contributing to higher percents of adipose tissue. And that's really important because dysregulating of these normal processes that govern the balance between epithelial and stromal tissue can lead to a loss of protection against epithelial proliferation and tumor development. So this, is I thought, really exciting findings to demonstrate, again, consistent with our work on those earlier measures of TDLUs, that PM2.5 does seem to be impacting the histologic tissues in the normal breast in a way that may predispose women to go on to develop breast cancer. So again, the story continues, and we're realizing more and more that PM2.5 components really can play an important role in how this exposure is related to breast cancer. And recently, we've really been thinking a lot about exposure to wildfires. As I'm sure many know on this, we're all experiencing the impact of more intense and increased exposure to wildfires. So this map shows wildfire smoke exposure across the US. This is definitely an exposure that is predominantly impacting the Western US, but is also traveling and impacting everyone. The wildfires as a source of PM2.5 is on track to be one of the primary sources of exposure to PM2.5, and indeed will completely reverse the gains that we've made in air quality after the Clean Air Act. And projections show that moving forward, that we'll actually see PM2.5 levels start to increase again. So despite the fact that wildfires are increasingly frequent and more intense, we know very little about the long-term chronic health implications of this exposure. And wildfire smoke has a unique chemical composition. It contains a relatively high concentration of mammary carcinogens, as well as estrogenic PM2.5 components. And this image shows the smoke from the wildfires in Canada in 2023 that we experienced in New York, how the wildfire smoke from Canada traveled to New York. And I know that here in North Carolina, we also experienced that exposure as well. So we're currently working with a collaborator, Tarek Benamarnia at UCSD, to link wildfire-related PM2.5 exposure to sister study addresses to evaluate associations with breast cancer. Given that this is such a new research area, there's a lot of, we're interested in a lot of different metrics of exposure. Wildfire PM is different than the regular air pollution that we experience because it's more sporadic. So we'll be exploring different metrics such as that incorporate frequency, duration, and intensity to better capture how wildfires might contribute to breast cancer risk. And the sister study is a great population to do that. This map shows the participants' residence at baseline, and you can see that we have participants across the US. So we'll have women in the Western US who are experiencing these more intensely, but we'll also be seeing women who experience lower levels of exposure if they live, for example, on the East Coast. So I actually have some preliminary data to share, which is very exciting. So we first started with linking this exposure metric among the sister study participants who live in California. So this is over 4,000 participants and 267 cases of breast cancer in this subset. And so we looked at these different metrics of exposure to explore, again, as I mentioned, how different ways to conceptualize how this exposure might be relevant. And so overall, we see that wildfire PM2.5 is not significantly related to breast cancer. But when we broke this out further by ER status, we see evidence of quite a strong association between mean wildfire PM2.5 and mean daily wildfire PM2.5 during peak weeks in relation to ER negative tumors. And here we are working certainly with a smaller number of cases in this preliminary analysis, but we see in general these odds ratios appear to be elevated across these different measures. So I'm really curious to see how this extends to the full cohort, which we're working on right now, and excited to share these results as they continue to come in. So with that, I've showed a number of different studies today, and data from large prospective US-based cohort studies, we do see that NO2 is associated with a higher incidence of breast cancer. And although this association is overall quite small, when we incorporate more long-term measures of exposure like we did in our historic model, we see that this is likely an underestimate, and then we start to see slightly stronger associations. For PM2.5, which is a more heterogeneous exposure, the story definitely appears to be more complicated. For breast cancer, we see that this relationship varies by geographic region, notably very strongly by tumor subtype, as well as by exposure timing. And our preliminary data suggests that wildfires may play an important role in ER-negative tumor development. Overall, I think this work has very important public health implications, given the fact that we are all exposed to air pollution, and this is very widespread, and that really we're on track to see a reversal in the gains that we've made, and that there will be wildfire-related changes in air quality, and not just in terms of increasing our total PM2.5 exposure, but increasing or changing the type of PM2.5 that we may be experiencing. So with that, I'd like to take a moment just to thank all of our collaborators across all these projects, in particular a postdoc fellow in my research group, Jennifer Ish, who's led a lot of this work, as well as the participants in the sister study, the Nurses Health Studies, and the WHI. I'd like to thank you all so much for joining us today, and I'd be happy to take some questions. I've also included my email here, so feel free to reach out if you have anything else, any other questions. Thank you so much.
Thank you so much, Dr. White. So, I'm going to invite our participants who are online to enter their questions in the drop-down menu where it says questions. I would ask that you don't use the chat feature. I know a lot of us get used to that because of Zoom, but if you could click on in the menu from your GoToWebinar, the drop-down menu for questions, you can type in your question there, and I will keep track of those as we go along. But I did want to ask you, and I know this sounds like a pretty rudimentary question for you since you're so immersed in this research, but what is the source of the NO2 that's leading to the higher incidence of breast cancer that is in our air, causing this air pollution?
Yeah, so NO2 is an interesting compound because we see it's related to breast cancer, but we also see it's related to lung cancer. But NO2 itself doesn't actually appear to have very, many carcinogenic properties. It doesn't appear to induce mammary tumors in animals in experimental work. So it's really considered to be a proxy for exposure to traffic. And traffic releases a wide range of compounds, polycyclic aromatic hydrocarbons, benzene. And so I think of NO2 as kind of a proxy for this mixture of pollutants that we're exposed to from traffic.
And so the PM 2.5, similar story?
So yeah, and this, is also, this is why air pollution, I think, is, a challenging thing to study because it really differs. So for PM 2.5, traffic is contributing, but we also see impacts from industrial emissions, and wildfires, and the contribution of sources varies in different places. So, we see this higher association among women living in the Midwest, and PM 2.5 in the Midwest has more industrial components as part of it, and we see more metals there, and so it does make it very difficult to study because it's a constantly changing exposure based on both geography and now time. We're seeing that as wildfires become more dominant, the composition of PM 2.5 is also changing that people are experiencing.
And so there would be a change in the concentration of these chemicals associated with outcomes from climate change.
Yeah.
So today you showed that there's definitely a relationship with breast cancer, and that breast cancer really differed by the type of air pollutant. What can you tell us about why these differences may exist?
By the types of the tumor types? Yeah, so hormone receptor positive tumors, we think of those, the ER positive or PR positive as well, depending on the characterization. These hormone receptor tumors are driven by estrogen. And so I think that's why we see the associations, more evident with NO2, that we might be seeing that that's acting through a different biologic pathway, one that's associated with endocrine disruption, whereas for the hormone receptor negative tumors, they're not sensitive to estrogen for growth. And so I think it's interesting that we see, this effect with PM2.5 in particular that's, 10 years prior. And to me, that suggests that this is acting more as a carcinogen, acting earlier in the process of initiating the tumor.
And that was an interesting concept for me of really thinking about the time, right? Because we tend to think of, cancer tumor development as being exposure and then the beginning of a process, a mechanism that leads to tumor development being much more confined in terms of the amount of time. But why do you think it takes from earliest exposure, is it have to build up over time, or is it that the cancer or the development of the tumor is actually just slower?
I think it could be multiple factors. There's the multiple hit hypothesis that it takes, multiple carcinogens, or, insults to result in a tumor. And then certainly tumors will grow at different rates. And, we know for breast cancer that, a woman's age at menarche, the time she gets her first period, plays a role in breast cancer development decades later. There's a whole, there's a lot of growing research on even exposures in childhood and infancy and how they relate. And so we know that, this is, as the impact of a diagnosis really can start very early, and the influences of the diagnosis. And, that's one of the things that makes breast cancer so hard to study, is we really need we need to understand long-term exposure histories.
So we have a question from one of our participants. If we increase oil gas drilling on land, will that increase the PM 2.5 levels in the air that lead to increased breast cancer rates?
It's possible. Drilling operations are something that we've been interested in and have been considering studying in this cohort as well, because there is information about where they're located. One of the challenging things about drilling operations is they come with a lot of different exposures. There's a lot more traffic. There is noise pollution often and light pollution from these, drilling areas, and there's a lot of stress that can be caused also by having these in communities. So I think that that's a really important research question that hasn't been well studied specifically for breast cancer, but certainly there are a lot of studies that have considered the adverse effects of living near a drilling operation, and I think increased exposure to air pollution is definitely one of them.
And I'm thinking about, with the concept of traffic, right? The volume of traffic seeming to be creating a higher concentration of these pollutants in the air seems to be the factor. So places where there's more cars, more traffic, higher amounts of exposure to these potential harmful chemicals.
Yep.
So how can people reduce their exposure to this if they're concerned?
I know, this is something that I think about a lot. It's really hard for an individual to change their exposure to air pollution. It's not reasonable always to tell someone to move your home, based on exposure to traffic. I think one of the best things we can do is, be aware of, different policies, and if we're concerned about something like air pollution, I think that's a policy, decision that, we want to advocate for policies that reduce air pollution for everyone. If that's, that I think that this is less of an individual decision, but certainly at the individual level, you can monitor air quality. There are ways to do that. You can choose to, not go outside on days for which the air quality is very bad. And so, you know, those are options. And certainly taking, you know, you can consider, you know, different air filters in your home, ways to reduce exposure if that's something that, you know, you want to do.
So, you know, we have all seen those air quality index reports on the weather, right? As part of our weather reports, and they tend to be at times of the year where it's a lot hotter, right? And obviously living in New England, right? Where it's the times of year when we're more likely to, you know, have windows open, have doors open.
The opposite in the
South. So if we're paying attention to the air quality index and we're seeing that there's an elevated risk, what are the steps to take? Like air conditioning, where there is already built-in filters. You mentioned air filtering. Is that through an HVAC system, HEPA filters? Like what investments will be helpful for me as a you know, someone living in or working in a more urban environment?
Yeah, certainly. So I am not at all an expert on the best types of air filters for your home, but I think, you know, that, you know, definitely something that you can find more information on the most effective ones. And, you know, I think it is important to be aware of, you know, how all these different factors could be influencing your risk of breast cancer. But I also think, you know, it's an important caveat here, right, is that the air pollution does seem to consistently be associated with breast cancer risk, but the increases in risk here that we're seeing in general are relatively small. And there are a lot of factors that influence, you know, an individual's risk of going on to develop breast cancer. And so, you know, this is a piece of information for people to consider. But I also think, you know, there's a lot of benefit for physical activity and being outside. And, you know, we have to balance these things, and that can be, you know, difficult to do on the individual level. So, you know, I think of this as, you know, information to help people make decisions, but I also recognize that for most people, you don't have as much control over this one particular exposure.
So you talked a lot about, you know, really the link between air pollution and breast cancer, and particularly the pollutant impacting the particular type of tumor development. What do you know about air pollution as it's related to other women's cancers?
Yeah, so we've actually been recently exploring that. So there's been very little study of how air pollution is related to other cancers. And recently in the sister study, we have observed that exposure to NO2, that marker for traffic, is also related to the risk of uterine cancer and ovarian cancer. And these associations do appear to be a little bit stronger than what we see with breast. So, you know, that's it still, you know, requires other studies to confirm this. And there's really not a lot of evidence out there, but there is some evidence to suggest that as well, air pollutants are related to gynecologic tumors in women.
Wow. So tell us a little bit more about this sister study. I think that, you know, longitudinal studies are usually, you know, have to be entered into with some great intent, right? Like because it is going to be, you know, data that's collected over, you know, throughout the course of a scientist's career, right?
Yeah.
So what was the impetus to initiate this sister study, and what information have you been gathering along the way, in addition to support for this link between air pollution and cancer?
Yeah, so the sister study is a very unique and special resource. So it was started in the early 2000s with really the goal of developing a large-scale, well-conducted study that could address a lot of different hypotheses. A lot of the work that had been done previously was focused on case control studies, which can be really difficult to, you know, take inference from for environmental exposures, because you're mostly focusing on kind of that time right around when, you know, your case was diagnosed. So we really really wanted this, longitudinal prospective study. And we've collected, just there, you can learn more on our website, sisterstudy.org, but, just a wealth of information about early life, pregnancies, breastfeeding, environmental exposures, really with the goal here to better understand how environmental exposures contribute to a woman's risk of cancer, but we also collect information on other outcomes. And we've done the work is, beyond just my research program. Certainly there's a lot of investigators that work on this study. We've published on nutrition, on physical activity. A lot of my work has also been focused on chemical hair products. So we've also been interested in that as a source of chemical exposure. And we've found that in particular, chemical hair relaxers have been associated with a higher risk of breast, ovarian, and uterine cancer. So a lot of that, those really key findings came out of the sister study. And we're doing more work. So we have, biospecimen on all these women. So I'm currently also leading some other studies where we're using large-scale environmental technology to measure thousands of chemicals in the blood samples and to try to look at signatures or profiles of how multiple chemicals might be related to breast cancer risk later on. But this study is very special. We just celebrated our 20-year anniversary, and because of the unique study design and that we recruited sisters of women with breast cancer, our participants are still very engaged. They care a lot about the study. Our response rates have stayed very high despite a lot of other studies having challenges. With COVID, we've really managed to keep our participants they're really proud of the study and proud of the work that's done. And we spend also a lot of time, communicating back with them about the findings that we've had. There's a very active Facebook group. And, it's just a real pleasure to work on this study. And it's been, just a fabulous resource, I think, for a lot of different research questions.
And you brought up the hair relaxers, and we've had other public health researchers as well as scientists speak to the chemical exposure associated with hair relaxers, which disproportionately impact women of color. Absolutely. And I guess, my question is, when we think of large urban areas, we tend to think about folks who are living in areas where there may be a higher level of diversity. And so, it seems like there could be the potential for like a concentration of exposures over lifetime. And I think, for us as individuals and survivors of breast cancer, we want to live our healthiest life. And we've had, conversations in Massachusetts Breast Cancer with other scientists around the idea that cancer is in fact an inflammatory disease. And so could you speak to that a little bit about the idea of how do we actually go about decreasing the potential of exposures that lead to inflammation that are impacting our health and may or may not necessarily lead to cancer, but definitely impact our overall health and quality of life?
Yeah, I think that's a great question and a challenging one. I think understanding, so getting to the inflammation point, I think part of that is, having really strong studies that are looking at, markers of inflammation and understanding how, in having well-conducted exposure assessment. I spent a lot of time talking about the work we put in to not just look at air pollution at one time point, but to try to do a better job of understanding a woman's, as much as we can. We can't go back for all of our women because there was not monitoring data. We can't go back much farther. So I think part of it is improving our science to better understand a woman's total exposure. And that's, a lot of the work that we're doing now in terms of the chemical biomarkers is, let's not just look at how your exposure to one chemical is related to these outcomes. Let's understand all of these chemicals, and it's not easy. We're working with thousands of chemicals, but how do you using new bioinformatic tools to try to take the more complicated nature of our lives into account. And we're working on it, and it's hard, but I think it's really important because we're not just exposed to air pollution, as I think you were suggesting. We're not just exposed to hair products, and we need to try to disentangle the fact that these exposures do sometimes occur together, and how do we best understand what's most important to target for interventions.
We do have a question about fragrance. Lasting,
Yeah.
Some fragrances last over a decade, They get into your clothing, right? Why are they lasting so long now? And how are they able to contaminate? How are they able to contaminate via the air and washing for so long? So the idea of fragrance, not just personal healthcare fragrance or fragrance that we may be putting on our body, but there's a lot of products that contain fragrance.
Right. So those are, often phthalates. So phthalates are the chemical compound that we typically associate with fragrance. Certainly, I think that that's true. Like the industrial processes to make these chemicals and the new generations of these chemicals are more, robust. So I think I personally do try to avoid, I try to find fragrance-free products. I think they're really challenging to study. The biomarkers that you measure in blood for fragrance are very short-lived. So it's very difficult to get a measure of long-term exposure. So it's not something that has been strongly found to be related to breast cancer. But I think that's because it's really hard to study. And certainly these have been related to other health outcomes. So, it's definitely challenging, and it's really a very, it's an exposure that also is very ubiquitous.
Well, thank you. That seems to be all the questions that we have for you today, Dr. White. On behalf of myself and the Massachusetts Breast Cancer Coalition Board and Cheryl Osimo, MBCC's Executive Director, I want to thank you, Dr. White, for this very informative discussion today. And I want to thank all of our many listeners for joining us. For those interested, the recording of this webinar will be made available later today on the MBCC website at mbcc.org. I would like to, if you would like to further support our mission of preventing environmental causes of breast cancer, please consider making a donation by visiting mbcc.org/donate. I appreciate your consideration. Have a good afternoon. And again, thank you so much, Dr. White.
Thank you, Dr. Malgos. Thank you so much for the opportunity to be here. Okay.